Okay, so by the time we're done with the Krebs cycle, we have about 4 ATP made from substrate level phosphorylation during the processes of glycolysis and the Krebs cycle. We also have a lot of these NADH molecules, and I forgot to mention them before, but the Krebs cycle also generates 2 FADH2s, which are similar to NADHs and basically act as electron carriers. So the next phase, the electron transport chain, is all about trying to convert the energy of these NADHs and FADH2s into ATP. So it's complicated, but basically, if we zoom in on the mitochondria, we have the outer membrane, and then we have the inner membrane here. And embedded in the inner membrane are a bunch of proteins, and I've simplified this quite a bit. But there's one key protein called ATP synthase. ATP synthase has a binding site for phosphate, a binding site for ADP, and it sort of wants to combine these two. The problem is, combining these two, as we've already seen, is endergonic. It requires energy. And so somehow we've got to couple the endergonic production of ATP here with something else that's exergonic. And that's what this whole setup is about. So it all begins with NADH. Okay? These are the NADHs from glycolysis, that transition reaction from the Krebs cycle, all of them. NADH finds its way into the mitochondria and it donates an electron to the electron transport chain. Now, that electron is going to be passed through a series of proteins, each of which is slightly more electronegative than the previous one. What that means is each one is holding the electron a little bit tighter than the one before. And so think of it like this is the top of the hill, and as the electron is passed, it's slowly going down the hill in terms of energy. Potential energy is being given off every time it's transferred. Okay, so as the electron moves, it's exergonic. Energy is being given off. That energy is coupled with the endergonic movement of hydrogen ions against their concentration gradient. And so as this electron is passed through these proton pumps, that movement is coupled with the pumping of hydrogen ions against their concentration gradient. You can see there's a lot more here than there are here, so it takes energy to go this way. As the electron is passed from this one to this one, again, it's coupled with the endergonic movement of H pluses against their membrane, and so forth. And so basically, all of these proton pumps are moving these against their gradient. When that electron reaches the end, it is actually passed to a molecule of oxygen, which is there because we breathe. That's where the oxygen comes from. And when the electron is joined with oxygen, a chemical reaction occurs that converts that oxygen into water. And so this is why oxygen is needed for cellular respiration and water is produced for cellular respiration. That step right there. Now, what's the point of all this? Well, guess what? These hydrogen ions are now piled up in that intermembrane space. So they want to get out of there. It turns out the only way for them to go back down their gradient is through ATP synthase. And since they're going down their concentration gradient, this movement is exergonic, which means it gives off energy, and that is coupled with the endergonic joining of these two together into ATP. And so through this electron transport chain, the cell is able to capture or convert the energy of NADH into ATP. And so here's a schematic drawing of the electron transport chain, and you can see here's ATP synthase. It's ready to put ADP and phosphate together into ATP, but it needs energy somehow. And so the way this works is just a big old game of energy coupling. NADH passes an electron to the proteins in the electron transport chain, becomes NAD+. That NAD+ is then used, recycled again in the Krebs cycle and glycolysis. The electron is passed from protein to protein to protein. ultimately being passed to oxygen to form water. And because each of these proteins is slightly more electronegative than the last, energy is given off every time an electron is passed down this chain, and that energy is coupled with the endergonic movement of hydrogen ions against their concentration gradient. So we get this huge buildup of hydrogen ions on the inside of the inner membrane space. Oh dear. And then those protons have only one way out, and that way out is through ATP synthase. And since now they're going down their concentration gradient, this movement is exergonic, and that is coupled with the endergonic production of ATP. So by way of summary, this diagram or this table right here kind of highlights the yield of these electron carriers from each of the major processes: glycolysis, that transition reaction, the Krebs cycle. It gives you the ATP yield. Note that it takes 2 ATP to shuttle NADH from glycolysis into the mitochondria. And then the ATP yield from oxidative phosphorylation, where each NADH gives you about 2.5 ATP. And so these two NADH ultimately give you 5 ATP, and so on. So you don't need to memorize these numbers, but certainly understand that you get a lot of these NADH, FADH2s in the Krebs cycle, and that the big payoff in terms of ATP is in oxidative phosphorylation during the electron transport chain. One other thing at the very start of this document, this is kind of a nice little overview that just kind of shows the major players and the inputs and outputs. And I think that's worthwhile to take a peek at that and make sure you understand all the major changes. But so long as you understand exergonic reactions drive endergonic reactions, that's it. The rest is just the details of the particular steps in the process. So focus on those big concepts, the big intermediates and the players, and try not to get too lost in all the details.